Afternoon, and you are welcome to this. I'm glad to see that most of the graduate students are here, and this will be quite pertinent for you. So, and I'm thankful to the organizers to give me this opportunity to share this thing with you. Now, there are a few terms that I want to be clear. Cowpea is a generic term for many varieties of cowpea, and this term was coined in US sometimes in 18th century because it was mainly grown for fodder as cowpea, and then it is a name that stuck to it. But each country has its own different name. Now this is black-eyed pea that in California and Texas you grow here. This is green, better green from southeast USA, South Carolina, Mississippi, and so on. But these other types are in other countries. This is called rough seed coat. This is a brown seed, but rough seed coat, white seed, rough seed coat, West Africa. This is in Cuba and many Caribbean countries, and red seed is popular in many other countries. So these are different varieties, but black-eyed pea is not just cowpea. It's one of the cowpea varieties. Now, plant breeding, to me, I think all of you know what is plant breeding. Plant breeding to me is improving the genetic makeup of plant, improving the genetic makeup of plant for human welfare. And this genetic makeup has two components now: the conventional breeding, which up to 1996 was the only method. After 1996 we got the molecular intervention. And conventional breeding is where you are making genetic improvement by phenotype basis. Phenotypic segregation in genetic population, you are making selection. And molecular plant breeding is where it is genotype based. You are manipulating the gene itself either through gene transfer or RNA intervention, and now the genetic editing and so on. An international collaboration is where the national and international agencies rise above their self-interest and unite together for a common cause. So these are the terms I thought we might as well understand as we go along. Before I tell cowpea story, I want to do some commercial for the International Year of Pulses. This is International Year of Pulses announced by the United Nations, and I don't know how many of you know what pulses are. Pulses are the food legumes which are eaten as dry seeds. There are many food legumes which are used as a fodder, vegetable, and so on, but the pulses are the group of food legumes which are eaten as dry seeds, and these are the common pulses. Beans, which are popular in Americas, cowpea in Africa, and these are Asian crops here, the chickpea, lentils, pigeon pea, and so on. This is our improved cowpea variety. This is bean variety, and I want to show you some others. You might not have seen lentils, mung bean in India, chickpea in Asia. Even now you are growing some chickpea here. This is field pea, urd bean, which is black gram, and pigeon pea. So these are the different pulses. The major difference between cereals and pulses is the protein content. The carbohydrate is more or less similar, ash content slightly higher, but mainly protein. Pulses are almost 2 to 3 times higher in protein compared to cereals and root crops. Also, the essential amino acids, in case of cereals, they lack in lysine and tryptophan. Pulses lack in methionine. So when we eat them together, we get the balanced diet. Now somehow nature wanted us to eat cereals and pulses together, because the origin of crops, when the continental drift happened about 65 million years ago, then the crop evolution happened in different continents. So in Americas, the maize, beans, peanuts, sweet potato, and so root crops, maize and beans were here. In Asia was chickpea, barley, wheat, pigeon pea, and soybean, and in Africa sorghum and cowpea. So you see there is a legume along with the cereal or root crop, so that early settlers, those of you who remember your history of the human settlement, early settlers settles in these places. In Mesopotamia and this part here in Africa, where there was pulses and cereals together, and that gave them the balanced food. So even now, particularly Asia, part of Africa and Latin America depend upon cereals with pulses and root crops with pulses. And this is what nature wants us. Now, some of the countries are too much dependent upon meat. And that leads to all kinds of problems. Now, if you replace one of these burgers from the double McBurger and take 1 cup of beans, your health will be very good. So that's what that means. So this International Year of Pulses is creating awareness that meat eating is good for you, but don't eat too much, eat some pulses also along with that. So that's the end of my commercial. Now we come to the cowpea. Now cowpea is a very versatile crop, and it is grown for many things. For vegetable, this is yard-long bean, and many places around the world it is very popular, mainly vegetable. Then fodder type as a forage crop. In East and Southern Africa, they eat leaves of cowpea like spinach. And actually, if you go to a small village, the housewives had 10-15 plants behind her yard, and then whatever water they wish, they put it on the cowpea. And these are very late maturing cowpea. They will pick the leaves third from the top and leave the tip growing, and they use vegetable like that. But most of the cowpea is used as a dry grain in various snack foods or regular foods and so on. So cowpea is a very versatile crop. A brief history of cowpea is known by different names in different places like black-eyed pea and so on. It originated in southern Africa, part of Botswana, South Africa, Namibia and other places. And then from there it is spread to other parts of Africa. And then it came to US through slave trade, from first in West Indies and then to southern USA. So many southern states grow it. And those who are from Texas, you eat black-eyed pea as a good luck on New Year's Day. And my message is, why not eat every day for a good luck throughout the year, so not just one day. Cowpea is currently grown over 80 to 100 countries. And here is a visual picture of where it is grown, over 80 countries, 14.6 million hectares, and about 7 million tons production. It is mainly an African crop. About 80% of cowpea is grown in West Africa, out of which 60% is in Nigeria alone. And this is map of Nigeria. I was based here in Ibadan, and finally then I was transferred to Kano. We had a station mainly for cowpea research, and that was set up when I went there. So I was based here for 17 years, 8 years here, and so I spent most of my time on breeding cowpea in Nigeria and West Africa. So it's a very important food crop, snack food, but this is how it is grown in Africa, as an intercrop with millet and sorghum. And you can see how widely distanced these have been planted, because there is no fertilizer. If a farmer plants it densely, nothing will come out of it. So by sparse planting, they get a few seeds. We had some students who worked on this. You get about 50-60 kilogram, less than a bag of cowpea per hectare, and 200-300 kilogram of cereals. That's all they have. So the varieties are spreading type, but grain and fodder both are very important in West Africa. There are major production constraints. This cowpea has high protein and virtually no anti-nutritional factors in leaves or seed, so it invites a lot of insects and diseases also. So you see there are several diseases, but 15 major diseases that affect cowpea. 6 major insects: aphid, thrips, maruca, spot bugs, curculio in Americas, not in Africa, and bruchids. Parasitic plants. You don't have parasitic plants here in the US, but in Africa they have Striga and Electra on cowpea and Hermantheca on cereals. Abiotic constraints, which is same for many crops: heat, drought, low fertility. And then the local varieties, the popular varieties all over the world, up to 60s, were late maturing and spreading type like this. So if grain and fodder both are important, and you cut 1 square meter from here, you will get very little grain and very little biomass. So how do we improve such a variety, for higher yield and so on? So I joined sometimes in 1979 IITA, which has a major crop. Before that, I was working on soybean in India. So Cowpea was an introduction to me, and I spent there almost most of my life, 27 years. So here are some of the constraints, biotic constraints. Nematode, this is bacterial blight, septoria, ascochyta blight, viruses, 8-9 types of viruses, and this is striga. You might not have seen, but striga is a parasitic weed. The seed germinates in the soil, attaches to cowpea roots, and draws the photosynthate from cowpea and comes out, and cowpea is virtually killed. So this is a very damaging kind of a parasitic weed in Africa. Then insects, this is aphid, which is here also. Thrips, they are also here. They cause a lot of flower drop. This is serious thrip damage. There is virtually all the flowers will be dropped. This is Maruca pod borer, which is in many other legumes also, a very devastating pest. And these are pod bugs. There are several types of pod bugs that suck the sap, and in the storage you have the storage weevil. So these are the major insects that affect cowpea. And these are the abiotic constraints. So you have drought. This is heat. When there is a hot temperature above 38-39 degrees Celsius, then the flowers drop. There is virtually no seed set. And then the low fertility. Most of the African soils are very sandy, 85 to 95% sand, no organic matter, very little phosphorus. So these are the abiotic constraints. So this is a kind of a progressive history of cowpea improvement. The systematic breeding program started in 1960s in some countries. Some genetic work was being done in US before. But IITA, International Institute of Tropical Agriculture, those of you who are familiar with the international setup, there are 15 international institutes around the world. IITA is in Africa, International Institute of Tropical Agriculture. Mike Thompson was with IRI in Philippines. So there are 15 of them. IITA was given charge of cowpea as a global mandate. They also work on many other crops. And I joined IITA in 1979. So IITA started work in 1970, and that was the one that stimulated a lot of work. So from 1970 to 80, the focus was on collecting germplasm. Now the strength of conventional breeding is the germplasm. If you have enough germplasm and you can screen it, virtually you will find everything. And that is what the story I'm going to tell you, how good this has been for cowpea breeding. Then the focus on seed type, striga resistant and drought tolerant, and up to now we have been working on low phosphorus, heat, and so on. And from 2006, we started interacting with molecular people to bring a Bt gene for Maruca resistance, and that's the story I'll be telling. So this whole process is still going on, but cowpea has moved from a crawling crop to one of the very high yielding type variety, and hopefully when the Bt cowpea comes in another couple of years, there will be a revolution. There are many people involved here. As I mentioned, I spent more or less my life there. I retired from IITA in 2006, and currently I have made arrangement to be 8 months here and 4 months in India. Both places I have some cowpea research project. I'll be mentioning a little bit what we are doing. I have a long list of names who have contributed, my colleagues in Africa, India, and many other places. But here I'm thankful to many people, particularly Dr. Baltensperger, Dr. Bill Payne. I don't know how many of you know. They were instrumental in bringing me here. Then Dr. Creighton, who is a cowpea breeder, he helped me initially. And after that, I have the colleagues, Dr. Hayes, Dr. Hang Bin Jan, Miping Jiang, and others who are helping us. There are several PhD students who have gone through our process and contributed many things. I don't know how many of you know Julie Roth, Laura Messer, and Tale Alexander, Brijesh Angira is still here. They have worked on some of these projects. So I will be now running through quickly, show you a lot of pictures. I will not spend much time on it, just to tell you the power of conventional breeding and the need for molecular breeding. So here is what the 15,000 germplasm that came, IITA is still maintaining that, and we got a lot of variability. These are the two varieties. This takes 150 days to mature. This takes only 55 days to mature. So that's the maturity variation. This is the peduncle length. You can combine this. This is a small one. These are the early maturity, completely erect type, completely spreading type. And you see the sink here from less than 5 inches to about a foot. So we have a tremendous variability in the germplasm. And just I want to digress a little bit. You know, most of the crops evolved over 50, 60 million years ago. And any mutation that you can think of must have happened during this period. Now, whatever had no selection pressure was survived. Those who were selected out, they didn't survive. So through the process of selection and mutation, many things that we see now in our germplasm collection have been sustained over the years. So if we have enough collection, we should be able to find any gene that we are looking for. And that's the strength of germplasm. So we got a lot of variability. Then for disease resistance, I mentioned 15 major diseases, and we got resistance to everything that you look for. So we have resistance to 8 viruses, and we did the genetics. There are varieties which are resistant to all the 8 viruses. This is striga, nematode, ascochyta blight, aphid, this is thrips, and bruchid. So we got resistance to most everything that you are looking for. But it's a lot of work. We had a lot of germplasm to screen. And to screen a germplasm, we have to create an environment where your phenotype is expressed. So if we are looking for ascochyta blight, you go to a place where ascochyta is endemic, where it is really, so we had to go to a hilly station in Nigeria where this disease comes, and I had 1,600 germplasm, only one survived. Striga, similarly, we go to a striga infested field, you plant the germplasm, and you have to have an environment, or either natural or artificial, to screen. And we found resistance to most everything that we were looking for. So here is the abiotic stresses. I think my cursor, yeah. This is a drought screening method that I devised when I was in IITA. When you go to field for drought screening, the soil moisture, the depth of the soil moisture, or the rain, so many things interfere. So we thought, why don't we bring droughts in the greenhouse and see how it works for cowpea. So these are wooden boxes. You plant your test lines. This is about 4 or 5 inches soil. And after they germinate, you stop watering and see which one dies first. No measurement of any physiological parameter or anything, just straightforward. And national programs really liked it. So this is soybean. It dies within 1 week after terminating the water. So maize survives 1 or 2 more days. But cowpea, there are varietal differences. Some will stay up to 70, 80 days without any water. Then you water it, it will regenerate. So we found a very simple method of screening for drought, and that has worked very well. Dave Verbery, who did PhD here, he did on this one, and it works beautifully. I will be showing some slides. This is heat tolerant. Simply grow the germplasm in hot air, hot weather. Now your June, July, August here is very hot. In Nigeria also, April, May, June was very hot. So we screen lines where you will get 0 yield versus no reduction at all. So we have complete resistance. Root architecture. This is deep and dense root system versus umbrella type. If rain stops today, 3-4 days this will get water. After that this will dry out, but this will continue getting water. So we combine this with this and you get super drought tolerant. These are all through conventional screening. I had 1600 germplasm line in a field that was drained of fertility for 2 years by planting maize continuously. Then we came here and we found beautiful genetic differences among the different lines. So this is Dave Verbery, who did PhD from here. He was with Dr. Bill Payne. And this is our box screening here. So you can see this line here. Even in the pot culture, it shows. And here is a line that survived up to 70 days. Everything is dead. And then when you rewater, this is 100% recovery. The one dominant gene. Just plain conventional method. Now when we published this, people didn't believe how can drought tolerant be one gene. But what we are doing here is removing the effect of root. Only the shoot dehydration tolerance is there. So once the plant senses the drought, that gene is turned on, closes stomata or adjusts the osmotic pressure and so on. Now this is the gene that has been discovered through biotechnology, and it is showing exactly the same thing in wheat and rice. So now people believe what we published was right. So this is just through conventional screening we found all these resistances. Heat tolerant, Brijesh Angira is not here, but he is our PhD student right now, and he has completed his thesis. And in 2011 was the hottest year in Texas, and we had about 43 lines, and we found that some of them were completely dropped flowers. There was no yield at all, and some were almost unaffected. And it was affecting the pollen. The susceptible lines, pollen development will be badly affected. And so this versus this, and it has come out to be one gene. I will be showing a slide later on. Julie Roth worked on low phosphorus tolerance, and Tully Alexander did his master's degree on this. And here is zero phosphorus, 2 varieties. We screened several varieties. This is one of the best, 0 phosphorus, and this is rock phosphate. In Africa there is rock phosphate which you can use, but rock phosphate is difficult to mineralize by many plants. So we had treatment of rock phosphate also. So you look at this variety, which is a local variety from Nigeria, no P and virtually no growth. But this variety, in repeated trials, with 0 P, something happens to it, and it is taller than even the regular P kind of a thing. And so Julie Roth did this, and it is one gene difference. Right? One major QTLC found, and it is one gene difference. So we found very good source of low phosphorus tolerance. Then Dr. Avika and his team, but even before when I was in IIT, we did some work on quality improvement. So we found a range of different types. So protein ranges from almost 18 to 32% protein. So you can raise protein content as much as you want. Then other minerals, complete range here. So we found complete range of quality traits within the germplasm. And this is a slide again from here, where the antioxidants are related to color. The colored seeds have more antioxidant than white seeds. So your black eye pea has very little antioxidant, but the red and black color, which is eaten elsewhere, is full of antioxidants. So we have to have a compromise there. Now some of the new varieties, these 2 varieties are released in India, released in many other countries, have up to 30% protein and a lot of iron and zinc. Genetic studies, I was blessed with the many universities around IITA. So students will come do research with us, and some work has been done here. So we did genetics of most of these diseases, insects, abiotic strengths, and again you see luck will have that most of these are simply inherited. Some of them are recessive, but most of them are dominant. So this is where a plant breeder, plant breeders should be developing a variety, but also should be publishing paper. Now, publishing paper for publishing sake, I don't believe in it, because you publish it and nothing happens. But if you go screen germplasm, get a particular trait, work out the inheritance, publish it, give a gene symbol, you are immortal. And that is how it should be. Not just that you do a genomic study or you do something, publish a paper, no one benefits from this. So my suggestion to breeders is have a good germplasm source, and if you have a trait that you are looking for, look for that in the germplasm. Once you find, design a genetic type of study, find out the inheritance, publish that, give a gene symbol, and that is a paper. That is a contribution that will be helpful. So this is what has given us many publications and through student work. So now plant type, I showed that this is the local type, and if you cut 1 square meter, you will get very little biomass. So how do we improve that? And both fodder and grain are important in cowpea. So here is the germplasm variation. We had complete erect type and complete spreading type, which are local type. So we cross these, and it segregates into, this is kind of a 2-3 genes responsible for this. So we select something which is taller, little bit taller, and then it goes on the ground and combine early maturity. So by the erect type, early maturity, intermediate growth type, we got lines which were highly erect yield type versus the dual purpose. So you can have grain as well as fodder. So this is what we call as a 60-70 day variety, and this is 75-day variety grain type. This is 75-day dual purpose. So now that plant type, which was in the previous thing here, has been completely changed to these types, and the yield was 3, 4, 5 times more compared to what we had. And this is all through conventional breeding. And over the year, here is another thing a plant breeder can do, no one else can do, and you can be proud of. When I joined IIT in '79, this was a variety from Nigeria, very high yielding but susceptible to everything. And one by one, we were able to put all these R's here, resistance, just by using different kind of a selection. So over time, now a new cowpea breeder doesn't have to go here. You use this, you have a good source of resistance in a good genetic background. So over the years, we were able to pyramid genes for resistance. Here is another advantage I had in Nigeria. This is our headquarters in Ibadan, and I was located here in Kano. And Niger Republic, across the border, we could cross. So we had a very good diplomatic relationship. So, and here is One, the petrol comes from Nigerian One here, where IIT has a station. 2,500 millimeter rainfall here, and 200 millimeter rainfall there. So I had 5-6 locations where you select your breeding lines, F6 lines you bulk, split into 4-5 groups, and just 1 or 2 rows plots at all the places at one time. But physically go, don't send the robots, don't send UAVs, don't send your technicians, go yourself and rate them. And the ones which are doing well all along, now you move with them. Then we distribute them worldwide. So I had these locations where the agroclimatic as well as disease pressure, insect pressure was quite different, and we tried to do the screening and then went through this. This is just, pardon me?
Kilometers. What's the spread of the distance there?
Okay, so from Ibadan to Kano is 1,000 kilometers. So from here to here would be about 1,500 kilometers, something like that. But every 200, 300 kilometers, the soil type, the rainfall, like 2,500 millimeter rainfall here versus 200 there. So as you go north, the rainfall becomes less and less. So these are the local variety from Nigeria, Danila and Aloka, and some of these new improved varieties. You can see the yield is over 2 tons, 2.5 tons, both the grain and fodder yield, because the plant type changed and so on. And IITA was working with over 65 countries. They were our national collaborators. So each year we'll make an international trial of the new varieties. Many centers still do this. And they send that trial to international collaborators with a field book, with a protocol and everything. They keep one copy, give us back another copy. We analyze the data. And based on this, they release varieties. So over the years, over the years, 40 different cowpea varieties have been released in over 40 countries. And this service international centers are providing. So the varieties came out. Now the cropping system, and this was another challenge that we thought we should start. This is the system in Nigeria and Niger. Very low population, but you can see this gentleman has worked very hard to remove the weeds. But there is no fertilizer, so the yield here is very low. Similarly, here is slightly better land, but it's still not much. So we reasoned out, if farmer has no fertilizer, no inputs, we cannot do anything. But if the farmer has a little bit of input, not much, just even a small amount, then by changing the varieties, we can make a difference. So we reasoned out, if we take this row and put it here, we take this row, put it here, so we have a window now, and put a little fertilizer on the sorghum and one spray in cowpea, and this is what happened. So I had a couple of PhD students who worked on this, and this is 2 rows of sorghum and 4 rows of cowpea. You put a little fertilizer on the sorghum and spray cowpea once or twice for maruca, and you get 300, 400, 500% more yields. And USAID was giving us some funds, so some of their big shots came, and they gave us a lot of money to follow this up. And we had some very good projects for almost until I retired. We started with 11 farmers, and we had about 300,000 farmers growing this. And this became a very popular method. So a little fertilizer and 1 or 2 spray of insecticide made all the difference. And we provided this as a loan for 1 season to farmers and then got it back from them. And many of these farmers, they broke their poverty cycle just within 2 years. So this was very, very successful program. Now after retirement in 2006, India eats a lot of pulses. Actually 80% of Indians totally depend upon pulses. They are vegetarian. But our pulse production has remained stagnant at about 13 million tons since 1951. So we grow wheat from November to March and rice from July to October. And in between summer, there is about 3 months of summer when nothing is grown. So I reasoned out, why don't I take my 60-day cowpea varieties and put here, which are heat tolerant and so on. So this is the work I'm doing in India. 4 months that I go there, this is what we are doing. And I have got some funds from international systems as well as national government is giving us, and we have already released 5 varieties of this. This is this in the background here is the Himalayas here. The Himalayas. So these are directly introduced varieties from Nigeria. You can see here, and they are working very well. So it fits very well. You combine wheat, water it, and directly seed it, and you get a good crop of cowpea. We have released these 5 varieties of different colors. These are fairly high in protein content, good in minerals and so on. So this project in India is coming up very well, and I am hopeful that we have about potential of 10 million hectares under this new system that didn't exist before. So it will be. And India has these different pulses I showed to you, the black gram, the green gram. So we have cowpea now of different colors. So it match the preference of whatever you want there. And they are making, based on African taste, different snack foods to popularize the cultivation of cowpea. Brazil used to grow cowpea only in the northeast Brazil, where the African Americans, African Brazilians were settled. But they found out that in Cerrado, where soybean is grown, there is enough moisture after soybean that they can grow a crop of 60-day cowpea. And actually, some of my varieties were released and very popular. There was a farmer who met me. I had gone there for a conference, who grows 10,000 acres of cowpea. He has 25 combines that he uses at one time, and they were very happy. So Brazil is now one of the largest producers of cowpea, and hopefully they will be coming up very, very fast. And this is what Brazilians have done it already. You see, this is a soybean crop they have harvested, directly planted cowpea. These are the soybean residue, and this is how soybean cowpea crop comes, and they combine, and they are exporting quite a bit of this to India already. Now here we are trying to do 2-3 things. First of all, try to map some of these genes, but also Texas used to grow 1 million acre in late 30s, early 40s, cowpea. Texas is still the largest cowpea producer, but it has gone down. Now what we are trying to say, this is a slide that Nitya Rajan has prepared for something else. You grow wheat from October onwards and harvest it May, June. There is another 2-3 months of summer where cowpea can be grown, and we have, this is 60-day cowpea in Vernon. Actually it matures in 60 days. You can, your field is ready to be vacated in 65 days. So we get about 2 tons of cowpea grain and 2 tons of fodder. You can see the wheat residue here directly seeded. So there is an opportunity to make double cropping wheat and cowpea wherever there is some rain, occasional rain, and if you have an irrigation facility. So this is what we are trying to do. St. Murray is trying to find out if we can have maize cowpea rotation either for the conservation purposes or the double. So there is a possibility of double cropping in most of these wheat growing Texas area. So what has happened over the year is 1961, the total cowpea production in the world was less than 1 million ton. And in 2009 it was over 6 million ton. Now it is 7.5 million ton. So about 800% increase in cowpea production. All the green revolution in cereals and others is about 200% increase, but the cowpea is 600. And I'm hoping that with this new cowpea varieties that are coming, this will be the legume of the future. So now, if the conventional breeding was so good, why do we need molecular breeding? And what I'm going to show here is conventional breeding has most of the answers, but not all the answers. So I showed you the slides of insect resistance for many things, but for maruca, this is a maruca moth that lays eggs on the buds of the plants, and these larvae come out and they bore in the pod as well as in the flower, and it drops, and this is what happens, 0% yield. There are a couple of lines which give you a few little bit thing here. So here is my best varieties. You spray twice for maruca and you get 2.5 tons, 2 tons yield. If you don't spray, the improved varieties are still better than the local, but see the reduction, almost 5 times reduction. So this is where we were stuck, and this I noticed in late 80s, early 90s, that we are making good progress, but there is nothing resistance for, and that is when the BT work was being done by Monsanto, and we had a visitor from Louisiana State University, Baton Rouge, who came and gave a lecture to us that BT gene is coming and it will be helpful. Actually, he said that if you give me cowpea, I will put BT and give it back to you within a few, and I gave him some. I gave him some, and nothing, we never heard anything from him. But 1990, but that stimulated us thinking, and I persuaded my DG, he persuaded the chair board, and we set up a biotechnology unit in IIT. We hired 2 scientists, and they started developing protocol, but nothing came out of it. So just before my retirement, 2006 I retired, 2004, I had a visitor from USAID, very, very important visitor. And I showed him this slide and these pictures. And I said, look, this is what is at stake. And this 2 spray is no big deal. Even BT cotton, if you farmers spray 15, 16 times, 12, 13 times. BT cotton you still have to spray 2 times. So 2 spray is no big deal, but for African farmers, it is a big deal. Chemical is not available, or many times it is adulterated or very expensive. So even this 2 spray was a bottleneck. So I sold this idea to USAID visitor, and fortunately with him, Monsanto's regional rep from South Africa was also visiting. And together they started discussing with the high-ups, and Monsanto agreed to donate the gene, BT gene for Africa. So my next slide is, before even we did that, we got the endotoxin produced by different genes, and this is available. So Cry1AB was killing the larvae much better than anything else. So these are the international organizations, many organizations that join hand. IITA, AATF is African Agricultural Technology Foundation, mainly supported by Rockefeller Foundation, USAID, Rockefeller Foundation, Bill Gates Foundation has joined now. This is Department for International Development of UK, University of Virginia, Purdue University. This is Australian Institute. Dr. T.J. Higgins was the expert of his good friend of mine, and he has already transformed peas with alpha amylase inhibitor. He knew how to transform beans, the peas. So we requested him to put the gene into cowpea. And these different people supported him. He got the funds, and this worked beautifully. In 2007, the whole thing started, and in 2008, late, he had already introduced the first BT gene in cowpea. And 2009, testing started in Nigeria, Niger, and Ghana. And this has gone on for now several years, and next year, hopefully, the BT cowpea will be released in Africa. It took 10 years. Sometimes you feel that molecular breeding is better than conventional because you can do things overnight. It took us 10 years, even though the gene was free, everything was done, because there are so many protocols involved. Even now I'm not sure whether the environmentalists will not influence African countries, although Nigeria, Ghana, and Burkina Faso and many other countries are ready to release it. But I hope that they let them release. But if it comes there, I'm expecting a surge in cowpea. So this is where molecular breeding is needed. So it is like, I sometimes say that our hands, plant breeder's hand is within this species. But now molecular breeding has extended our hand anywhere in evolution. We can bring a gene and put it there. But we should not go at random anywhere. We just go purposefully what we need. And that is what the message I want to convey, that we needed a BT gene to control Maruca. And it was available and it has worked. Monsanto is considered as a monster company, but here it became really compassionate and it worked. And I hope that Africa benefits from this. Now in India, my program, when we grow cowpea in rainy season, Maruca and all kinds of pod borers are coming. So I'm looking forward to BT release in Africa, and I will bring that to India, and India will have a revolution in cowpea production. So this is a message I want to say, that we should not be just fascinated by molecular breeding, but use purposefully where we need it. And that is where the real need for molecular breeding is. Now here in Texas A&M, my purpose was to help Texas grow more cowpea to export to India and Africa and other places. But also I had this bunch of genes that we could get it from cowpea, identify it, and use it in breeding in other crops. So I had struggled to many scientists to convince, and finally first we had some students through Dr. Dirk and others did the genetics, and for last 3-4 years Dr. Zhang and Meiping Zhang have been helping us to develop mapping population, and we have gone far with at least one heat tolerance with Brijesh Angira, and you already know Zhang and Meiping Zhang, and they are helping us out. And here is the first thing that that come out, and it is, I mentioned that heat tolerance is one gene, and one major QTL came out from this. Actually, you look at the reels, and you see the segregation, it is half. Half of these F7 lines are tolerant, half are susceptible. So it's a major gene, major QTL. And Dr. Zhang says that he already has a marker, and it is just kind of a single SNP that he has found it, and hopefully will use it for marker-assisted selection. We have this mapping population for aphid, for heat, for drought. Hopefully we will be able to map this. Now imagine if heat tolerant gene is cloned from cowpea, put in wheat. Right now we are talking about heat tolerance in wheat due to wax coating. It's a mechanical thing. Here is a real genetic thing. So these genes will be very, very, very useful. And I hope we get some funding to get these genes out from cowpea. But even more fascinating is this slide here. When we develop mapping population and you go to F6, F7, F8, your probability of getting a new genotype is much higher. Supposing a trait is controlled by 4 genes. In F2, probability of getting the genotype, homozygous genotype for 4 genes is 1/4 to the power 4, 1 out of 256. But if you go to F6, F7, it is 1/2 to the power 4, 1 out of 16. So here is our F7, we got a 45-day cowpea and about 120-day cowpea when the two parents were 70 and 75. So here is the two parents are here and the two extreme genotypes are here. In F2 we did not get it because the probability of getting, so the mapping population, which is also you call single seed descent method, SSD breeding method, if you have large number, 5 to 6, 7, 800 reels, you can get some new genes that were never discovered before. So we are talking about 45-day cowpea. And I have been talking to Dr. Zhang and other molecular biologists, and Mike Thompson is here, that you are working on Arabidopsis, which is not a crop. We have a crop now which is 45 days. Arabidopsis takes about 30, 35 days and so on. Here, and you have a lot of genes that are here. So why not try cowpea as a molecular genetics crop in addition to our Arabidopsis? I'm not saying you do it. So what I'm saying is through this mapping population, which is the SSD method of breeding, if we have a large number of lines, we will discover some new genotypes that you never got in F2 and F3. So this is the maturity we have got, and here is the seed type. This is GEC, this is this type of eye, and this another one, this type of eye. F1 is self-color, and in F2 we got 2, 3 new seed types which never existed before. So we can get some very good genotypes, new genes discovered from mapping population. So mapping population helps getting the gene of particular interest, but also it gives us opportunity to have many genes, new genes that come out. So we have used African varieties that I brought from there and crossed with American varieties, Texas varieties, and these are some of the lines. We had a project in South Africa for 5 years, and some of these new lines, I'm calling them next generation cowpea varieties, which mature in about 65, 70 days. Yields are about 2.7, 2.8 tons. These couple of varieties have already been released in South Africa, and we are hoping to describe here in Texas also to be released. So there is a tremendous scope of improving a crop through conventional with what we call as a need-based molecular intervention, and we can do wonders. You know, plant breeding is a field that you can do magic. And I wrote a book 2 years ago where Crop Science published it. Cowpea is the future legume because it is 60-day varieties. It can fit anywhere. There is a 2 months period, and with all these protein and other things coming, whereas all other legumes mature 120 to 140 days. They compete for land. So I am expecting that cowpea will become the important legume in coming decades. And Texas state can benefit tremendously. Right now the focus is more on big ranches, meat production, but 30, 40 years from now, when world population goes up and there are vegetarians all over, the demand for pulses will be very high. And I think Texas should try to grow a little bit more, at least in the niches, double cropping wherever it is possible. So this is a story I wanted to tell you. Again, I want to mention here is that we get fascinated by the advances in biotechnology. Now people talk about genomics. Now gene editing has come, but gene editing mutation came in 30s. Muller got a Nobel Prize in mutation. People thought we can solve all our problem now by mutation. What have we solved? A few male strile genes, a few things have come out from there. Cytogenetics came, and Kihara and Sears and others mentioned that we can now take a whole chromosome from here, put here, and solve all the problem. How far have we gone? But those are the tools. Similarly, the molecular biology, the gene transfer, the RNAi technology, even the gene editing is a tool, and we can definitely make use of it. But let us not get carried away that your training in conventional breeding is jeopardized because you are fascinated by the molecular. So what my message is, conventional breeding is the main river of breeding, plant breeding. These mutations, cytogenetics, biotechnology are tributaries contributing to the river flow. And if we understand that, our training in conventional breeding should be the top. And still, we should have the molecular breeding training so that we can take advantage of both, but not one at the cost of the other. So that's the message. Thank you very much for the opportunity for me to have time for some questions or comments that you may have. Yes.
I have a question. In a situation in Brazil where you have cowpeas after soybeans, do you think there's a sharing of the bacteria that fix nitrogen between the soybeans that have the bacteria will transfer that to the cowpea, or there's no interaction between the two? And how is disease pressure on the cowpea after being a soybean field?
You're talking about rhizobium for soybean?
Rhizobium.
Yeah. No, these are two different. The rhizobium for soybean is different than the cowpea.
So there's no benefit?
No benefit. But what happened, cowpea is what we call as a promiscuous kind of a nodulating type. You take cowpea anywhere in the world, and any local bacteria will nodulate it, except for a few places. So, but you inoculate once or twice, 2 years or 3 years, and then takes over. So there is no competition there. Fortunately, the diseases between soybean and cowpea are not so common. So that doesn't build up, as you go along. And as I mentioned, most of the improved varieties are resistant to major diseases, only occasionally. Brazil is really running with cowpea and soybean. And soybean and cowpea both are introduction from IITA to Brazil. We had 2 breeders there in late 70s and early 80s who introduced this. But Brazil would move so fast we didn't imagine. And Brazil used to produce about 200,000 tons of cowpea. Now it's about close to 1 million tons. And they're moving even much faster. Yeah.
You mentioned that because it's a short duration crop, it can be grown during fallow months if you have 2 months gap. I wanted to know if it is photoperiodic sensitive and it would affect its use in different regions in the world for such pepper.
Very good. I missed the photosensitivity part of it. Certain cowpea varieties are photosensitive, which means in long day they will not flower. They will flower only in short day. So what these 60-day cowpea varieties are heat tolerant, photo insensitive. So no matter when you plant, but they are not cold tolerant. So in winter months, even in India, northern India, in winter months not, but in central and south India you can plant cowpea throughout the year. Wherever there is a niche. Now let me emphasize again, all other pulses that I showed to you, they mature between 110 to 150 days. So they compete for land with cereals. Now India and many other third world countries, land is limited and cereals are more producing than the pulses. So food security, the farmers don't want to, they want to plant cereals, So, but wherever there is a little gap between 2 cereals or root crop, we have a project in Nigeria where cassava is grown 1 meter apart. Sugarcane is grown 1 meter apart, and they are slow growing crop. You put an intercrop of 60-day cowpea. Before even that grows up, you harvest it. So there are many opportunities where the 60-day cowpea will come. So pulses production we are organizing a symposium in Crop Science Society, and I am making a presentation about the pulses production in world is also stagnant since 1961 because it competes with the cereals, and no one wants to grow pulses in place of cereal. Only Canada, Australia, and US, where you have surplus of wheat, part of the land now they are giving to pulses to export. India exports about 4-5 million tons of pulses each year, mostly lentils, chickpea, and peas from Canada, Australia, and US. So these countries, and even my recommendation is these countries should produce more, because 25-30 years from now, India will not even have any pulse in the international market to import, and India is really concerned. And if program like mine, that between wheat rice, rice, you can grow it. So that is where the short duration pulse comes into picture, and it can grow anywhere. It should have a little water. I mean, 2 months it is very fast, but still you need some water to grow it. So that is the advantage of this. Yeah. Yeah, I think you had
2 questions. One, the same one I asked you last time you gave the presentation. Why do we call them pulses? What's the origin of the word? And 2, would you comment, given that in cotton we now have resistant weeds to Roundup, and we have bollworms resistant to BT genes, would you comment on whether or not GMO is a failed technology?
Yeah, good. Both good. Pulses, I don't know where it originated.
You still don't know.
I still don't know. I had forgotten that you asked me. I should have done some research. But pulses must be of Indian origin, because everyone says pulses, and United Nations gave it a legitimate definition. That dry seed, and most of the pulses in India we are using dry seed. So might be Indian origin. And actually your pulse, so the food legumes are like your pulse
Keeps
for
our
life.
Hearts
Keeps you
What is dear to your heart?
keeps you going. Yeah, keeps you going. The other question about the breaking down of BT is no different than breaking down of any other genes. So we know that wherever we have a major gene inheritance and the pathogen is sexually producing many types, there will be a type that will come out which will circumvent the genes that is resistant. That is there. Now in most of the cases that I have aphid resistant and so on, it is discovered long ago. Fortunately we have not got this, but that possibility is there. So my take there is, take the benefit as long as it is there. Because right now Maruca is killing everything. We are spraying 2-3 times, 4 times. Africans don't have it. Suppose BT cowpea comes and they start growing all over Africa. Then 10 years or 5 years, 6 years later, they know, at least for 6-7 years they have benefited. What's wrong with that? That's my take. But if we are careful and follow some of those guides that to how to manage the resistance, it will work. So I'm not that much worried about this. Like BT, whatever BT cotton, it was released in 96. This is 2016. 20 years? Is it 20 years now? 20 years farmers have benefited. Even now Indian farmers are all for it. So occasional problem comes, it is no worse than what it was before. Now we have a weed, like even Monsanto is coming with another chemical that has its own complications. But we should be prepared to take care of something comes up. But in the meantime, if there is an opportunity, why not exploit that opportunity as long as it lasts? So that's my take. I don't know whether you agree or not, but right now the focus is more on big ranches, meat production, but 30-40 years from now, when world population goes up and there are vegetarians all over, the demand for pulses will be very high, and I think Texas should try to grow a little bit more, at least in the niches, double cropping wherever it is possible. So this is a story I wanted to tell you, and again I want to mention here is that we get fascinated by the advances in biotechnology. Now people talk about genomics. Now gene editing has come, but gene editing mutation came in 30s. Muller got a Nobel Prize in mutation. People thought we can solve all our problem now by mutation. What have we solved? A few male style genes, a few things have come out from there. Cytogenetics came, and Kihara and Sears and others mentioned that we can now take a whole chromosome from here, put here, and solve all the problem. How far have we gone? But those are the tools. Similarly, the molecular biology, the gene transfer, the RNAi technology, even the gene editing is a tool, and we can definitely make use of it. But let us not get carried away that your training in conventional breeding is jeopardized because you are fascinated by the molecular. So what my message is, conventional breeding is the main river of breeding, plant breeding. These mutations, cytogenetics, biotechnology are tributaries contributing to the river flow. And if we understand that, our training in conventional breeding should be the top. And still, we should have the molecular breeding training so that we can take advantage of both, but not one at the cost of the other. So that's the message. Thank you very much for opportunity for me to learn. Yep. We have time for some questions or comments that you may have. Yes.
I have a question. In a situation in Brazil where you have cowpeas after soybeans, do you think there's a sharing of the bacteria that takes nitrogen between the soybeans that have the bacteria will transfer that to the cowpea, or there's no interaction between the two? And how is disease pressure on the cowpea after being a soybean field?
You're talking about rhizobium for soybean?
Rhizobium.
Yeah. No, these are two different. The rhizobium for soybean is different than the cowpea.
So there's no benefit?
No benefit. But what happened, cowpea is what we call as a promiscuous kind of a nodulating type. You take cowpea anywhere in the world and any local bacteria will nodulate it, except for a few places. So, but you inoculate once or twice, 2 years or 3 years, and then takes over. So there is no competition there. Fortunately, the diseases between soybean and cowpea are not so common. So that doesn't build up, as you go along. And as I mentioned, most of the improved varieties are resistant to major diseases, only occasionally. Brazil is really running with cowpea and soybean. And soybean and cowpea both are introduction from IITA to Brazil. We had 2 breeders there in late 70s and early 80s who introduced this. But Brazil would move so fast we didn't imagine. And Brazil used to produce about 200,000 tons of cowpea. Now it's about close to 1 million tons. And they're moving even much faster. Yeah.
You mentioned that because it's a short duration crop, it can be grown during fallow months if you have 2 months gap. I wanted to know if it is photoperiodic sensitive and it would affect its use in different regions in the world for such preference.
Very good. I missed the photosensitivity part of it. Certain cowpea varieties are photosensitive, which means in long day they will not flower. They will flower only in short day. So what these 60-day cowpea varieties are heat tolerant, photo insensitive. So no matter when you plant, but they are not cold tolerant. So in winter months, even in India, northern India, in winter months not, but in central and south India you can plant cowpea throughout the year. Wherever there is a niche. Now let me emphasize again, all other pulses that I showed to you, they mature between 110 to 150 days. So they compete for land with cereals. Now India and many other third world countries, land is limited and cereals are more producing than the pulses. So food security, the farmers don't want to, they want to plant cereals, So, but wherever there is a little gap between 2 cereals or root crop, we have a project in Nigeria where cassava is grown 1 meter apart. Sugarcane is grown 1 meter apart, and they are slow growing crop. You put an intercrop of 60-day cowpea, before even that grows up, you harvest it. So there are many opportunities where the 60-day cowpea will come. So pulses production, we are organizing a symposium in Crop Science Society, and I am making a presentation about the world. The pulses production in world is also stagnant since 1961 because it competes with the cereals, and no one wants to grow pulses in place of cereal. Only Canada, Australia, and US, where you have surplus of wheat, part of the land now they are giving to pulses to export. India exports about 4-5 million tons of pulses each year, mostly lentils, chickpea, and peas from Canada, Australia, and US. So these countries, and even my recommendation is these countries should produce more, because 25-30 years from now, India will not even have any pulse in the international market to import, and India is really concerned. And if program like mine, that between wheat rice, rice, you can grow it. So that is where the short duration pulse comes into picture, and it can grow anywhere. It should have a little water. I mean, 2 months is very fast, but still you need some water to grow it. So that is the advantage of this. Yeah, I think you had
2 questions. One, the same one I asked you last time you gave the presentation. Why do we call them pulses? What's the origin of the word? And 2, would you comment, given that in cotton we now have resistant weeds to Roundup, and we have bollworms resistant to BT genes, would you comment on whether or not GMO is a failed technology?
Yeah, good, both good. Pulses, I don't know where it originated.
You still don't know.
I still don't know. I had forgotten that you asked me. I should have done some research. But in pulses must be of Indian origin, because everyone says pulses, and United Nations gave it a legitimate definition. that dry seed, and most of the pulses in India we are using dry seed. So might be Indian origin. And actually your pulse, so the food legumes are like your pulse for life. Keeps you
dear to your heart.
Keeps you going. Yeah, keeps you going. The other question about the breaking down of BT is no different than breaking down of any other genes. So we know that wherever we have a major gene inheritance and the pathogen is sexually producing many types, there will be a type that will come out which will circumvent the genes that is resistant. That is there. Now in most of the cases that I have aphid resistant and so on, it is discovered long ago. Fortunately we have not got this, but that possibility is there. So my take there is, take the benefit as long as it is there. Because right now Maruca is killing everything. We are spraying 2-3 times, 4 times. Africans don't have it. Suppose BT cowpea comes and they start growing all over Africa. Then 10 years or 5 years, 6 years later, they know, at least for 6-7 years they have benefited. What's wrong with that? That's my take. But if we are careful and follow some of those guides that to how to manage the resistance, it will work. So I'm not that much worried about this. Like BT, whatever BT cotton, it was released in 96. This is 2016. 20 years? Is it 20 years now? 20 years farmers have benefited. Even now Indian farmers are all for it. So occasional problem comes, it is no worse than what it was before. Now we have a weed, like even Monsanto is coming with another chemical that has its own complications. But we should be prepared to take care of something comes up. But in the meantime, if there is an opportunity, why not exploit that opportunity as long as it lasts? So that's my take. I don't know whether you agree or not. But if someone says, don't go for vertical resistance because it will break down. Hell, we'll see when it breaks down. But as long as it is working, I'll take advantage of it. That's my take.
All right, and we gotta stop for now. Thank you, Dr. Singh, for coming in today. Let's give him a hand.